Hi, hello. We have started our survey on fungi, and this is the first life cycle on fungi, that is Rhizopus fungi. We can see that this is commonly known as bread mold. You can see a white cottony appearance on the surface of the bread. Why you are seeing this black color is because the tip of this particular spore is called sporangium, which is bearing black color spores. That's why it is black in color or gray in color here. But when we are looking to the microscope, you can see this white colored strains. So this is the mycelium. It is like a white cottony tuft, and on the tip of which you can see a pinhead type sporangia. We will see about this in detail in the upcoming slides. So in the surface of the bread you can see the Rhizopus fungi growing here. So let me remind you one thing. From this lecture to the upcoming lectures till we finish the fungi, I'll show you these slides, these 1, 2, 3, these 3 slides, because this reminds you that you need to cover all these topics to completely finish the fungi in entirety. So this is the first area that we are touching today, that is the life cycle of Rhizopus. So life cycle of Rhizopus comes within the phylum Zygomycota, and in the Zygomycota, what is Zygomycota, what is Ascomycota, Basidiomycota, etc. we have seen in the very first lecture. If you have not watched that lecture, I will strongly recommend you to first finish the syllabus decoding of fungi, then come to this lecture. Okay, so after Zygomycota, then we will be seeing the Ascomycota, and we will see all these things. Then we will move to Basidiomycota, then Oomycota, lichens, and the other general topics. So let us start with Rhizopus. I'll be moving in a bit faster pace in this lecture because I want to finish all these things, okay, from systematic position until economic importance in this slide. So wherever you want, you can rewind or pause for taking lecture note or for writing or copying the diagrams or notes which are given in these slides. So let me move. Okay, so you can see that this is the bread mold, and a commonly known and very famous bread mold of the Rhizopus genus is Rhizopus stolonifer. Okay, you can see this structure. The mycelium is white in color, and the tip which is swollen is called sporangium, that is black in color. So habitat, mostly it is terrestrial in habitat. Don't believe that it is only seen on the top of bread. No, it is even seen in soil. You can see this white colored mat. Then it is also seen in plants and in animals. Okay, it belongs to Zygomycota. So why this name? Because it will be forming zygospore during the sexual reproduction. We will see that. Then this thing we have already explained. Now vegetative structure on Rhizopus thallus. Of Rhizopus thallus, how you can see this colored matty appearance on the surface of the bread. So thallus is a white colony. Okay, this thallus is a white colony. That is called not colony, thallus is a white cottony mycelium. I'm sorry, like it's like a cotton mat, right, and are highly branched. So this is the thallus structure. We should understand that when it is germinating, all the thallus will be looking alike. Okay, it will be everything will be looking similar. There won't be this kind of divisions of sporangium or rhizoids like that. Everything will be similar, but as it matures, there will be 3 types of thallus differentiation. One type of thallus will be growing underground, that will be forming rhizoids, which are root-like structures which are present on the fungal body. Then there will be prostrate thallus called stolons, from which there will be a growth of erect branches called sporangiophores. Okay, the sporangiophores at its tip will be forming the sporangia, which are the reproductive structures. And you should know that you can see that wherever these rhizoids are formed, at these junctions, from these junctions, this particular sporangiophore is formed. Okay, this thallus, this erect thallus is called sporangiophore because this is bearing sporangium. This is sporangium bearing branch will be called sporangiophore. Okay, so these are formed. We are drawing this at the junctions where rhizoids are formed, right? So we are not drawing the sporangiophore here because there is no rhizoid here. So you should keep that in mind. Now, So about vegetative structure, I have already discussed everything. Let me see if I have missed anything. Yeah, you should understand that it's the thallus. It's like a tubular or non-septate and cenocytic hyphae. Okay, you can see that it is tubular, right? It's like a cylindrical or tubular structure, and it will be having throughout nuclei with no septa within. Can you see any septa? No, it is an aseptate condition. And cenocytic. Cenocytic means what? Many nuclei without forming any septa. So the cell wall will be formed of chitin chitosan complex. It is not cellulose, but it is formed of a polysaccharide called chitin and chitosan. Both are there, and other things I have already explained. Now rhizoids, like we know that it is root-like appendages. Then it will be penetrating into the substratum, so that will be fixing the mycelium on the like substratum. It will be secreting, these rhizoids will be secreting digestive enzymes into the substratum, and that will be digesting the food. For example, if it is a bread, or if it is an animal surface, if it is a fruit surface. For example, on tapioca you will see like the rot disease in tapioca. That is because these rhizoids are secreting some digestive enzymes to digest the like fruity part of that particular like potato, the thing that we eat. Okay, that particular area will be digested by these rhizoid enzymes. So then you can see that these clusters of long unbranched erect fertile hyphae, this is called sporangiophores. You know that there are vegetative hyphae and like fertile hyphae. Vegetative hyphae means stolons, which are prostrate hyphae, and rhizoids, which are root-like hyphae. These both are actually stolons and rhizoids are vegetative hyphae, and this unbranched erect hyphae which is bearing sporangium, that is called fertile hyphae, that will be bearing sporangium at its tip during maturity. We will see how sporangium is formed. Let us move to reproduction. Okay, there are three modes of reproduction. Vegetative, that is through fragmentation. You can see that like maybe due to a herbivore biting, or due to accidents, or due to drying up, a particular piece will be broken away, and that will be forming into new thallus. So that is called vegetative fragmentation. Then asexual and sexual. You can see these are the two modes of asexual reproduction. One is the formation of chlamydospores, and one is through the formation of sporangium. Okay, we will discuss this in the upcoming slides. So this is the kind of depressing images that we are seeing in the textbooks and all. Don't worry, I have attached very good colorful and easy to understand images in all these slides, so you can ignore these kind of depressing images. So asexual reproduction by chlamydospores. You can see that during unfavorable conditions, the protoplasm with like not the extreme protoplasm, you can see that maybe this is the tip, this is the top anterior tip, and this is the posterior tip. In between this area, the intercalary part of the protoplasm will be aggregating together, and that will be forming a thick wall around itself, and that will be developing into, that will be like developing a septa on both sides, and that will be converting itself into chlamydospores. Okay, so this will be detaching. So this was the fragment. Now it got detached from the thallus when the thallus dries up, and it will be laying in the soil till the favorable condition arises. When the favorable conditions arises, it will be forming the germ tube, and finally it will be developing into a normal thallus or hyphae. Now asexual reproduction through sporangiospores. Very simple. You know that this was the vegetative hyphae which got developed from a spore. Now there is fertile hyphae called sporangiophore, then like vegetative hyphae called stolon, and rhizoids. Now you can see that as the fertile hyphae matures, at the tip there will be, the apical part of the hyphae will be slowly becoming swollen. How? The entire nucleus and cytoplasm will be accumulating. Maximum part of nucleus and cytoplasm will be accumulating in this part. So this will be becoming swollen. Now once it is becoming swollen, there will be one differentiation in the tip part. There will be a peripheral area. You can see this greenish swollen part was not present before. Now this particular sporangial area got differentiated into two. A peripheral area which is having maximum nucleus and cytoplasm, and there is a swollen area like below that. So this swollen area below that, which is having minimum nucleus and maximum vacuole spaces, this area is called columella, and the remaining swollen area will be developing as a sporangium. Okay, Now you can see that this sporangium was having many nuclei and cytoplasm. Imagine that this is a cake, okay, the cake, birthday cake, etc. that we buy, and imagine that you are cutting that particular cake into many small pieces. Now you can see that each of the small pieces of that cake will be having many nuclei. Okay, the entire protoplasm we have cut into many small pieces, and each of the piece will be having many nuclei and cytoplasm within it. Now they will be developing a wall around itself, so that will be forming sporangiospores. Okay, now the spores will be like maturing within this particular sporangium. When the maturity period arises, it will be bursting. This sporangial wall will be bursting, and sporangiospores will be released into the environment. That will be forming a germ tube, which will be finally forming the vegetative hyphae. So I hope everything is clear. So the sporangiospores are developing inside the sporangium. They are multinucleated. Why? The cake piece had many nuclei, right? And they are non-motile. Okay, fine. Now let us move. This is the knot. Okay, I have explained everything in this knot in the previous slide. So this is a similar image. This is taken from the textbook. So, but you can follow this image. Okay, fine. Now sexual reproduction in Rhizopus. So this is also extremely simple. Maximum species of Rhizopus are actually heterothallic. That means there will be two types of thalli. One particular like thallus will be positive, and the other thallus on a different hyphae or a different thallus will be negative. So that will be coming together. Okay, compatible hyphae which can come together, that will be developing a small outgrowth. You can see they came together, this area became swollen, and this is consider this as a small outgrowth on the hyphae by like swollen apex. Now this will be known as, this swollen part will be known as progametangia. It is given as progametes, but that's not proper terminology. These outgrowths are called progametangia. Now in the progametangia there will be septa on the two sides. Here on the back side and on the front side there will be septa, and this septated, this small body which got developed at the tip is called gametangia. This gametangia will be having a lot of cytoplasm and nuclei, and after this thing, this particular septa that is in between this gametangium, that got dissolves, and it will be forming a spherical body. Slowly it will be converting into a spherical body that is called like a zygospore. But before this formation of zygospore, you can imagine that when this particular septa is dissolved, there will be many nuclei of different strains coming together. That will be two, or like just two out of this many nuclei will be fusing together. One nuclei from the plus thalli and one nuclei from the minus thalli. That will be forming the zygote. The entire remaining nuclei will be degenerated, and this zygote will be developing a wall, a thick watery wall around itself. Okay, that will be like developing a wall, and it will be, now it will be called a zygospore. And from this zygospore will be detached, and that will be laying on the soil till the favorable condition arises. When the favorable condition arises, there will be the germination of zygote into a promycelium. Why promycelium? Because this will be maturing itself into a proper mycelium. Okay, now this promycelium will be carrying a sporangium on its tip. Okay, on its tip, this particular sporangium will be having many like this particular, like we know that this is germinated from the particular zygote. So this particular sporangium is 2N. Okay, this is also 2N. Now it will be undergoing meiosis. Now it will be undergoing meiosis, and it will be releasing meiotic spores or meiospores. Okay, meiotic spores or meiospores. Okay, let me see if it can be extended like this. Okay, it will be forming meiosis occurs, and it will be forming haploid meiospores, and that meiospores will be germinating, and that will be forming into a new mycelia. Consider this particular promycelium as a sporophyte, just for the understanding sake. This was a gametophyte because these two were gametes, gametophytes. So from this gametophyte, this sporophyte aroused, and this on this sporophyte, from the like meiosis happens, and the spores got released. That will be developing new gametangium, okay, or new thalli or new gametophytes. In that way also you can remember. Now this is the note for the sexual reproduction. I have explained everything. You can pause the video and write it. Now this is the same figure which I have explained. This is just taken from a different source, just for a proper understanding, just in case if you want an extra clarity. These are easy to draw figures in the examination. Now this is the figure for the life cycle. If you want to draw, you can know that, okay, like if you want to draw asexual reproduction from the sporangia, spores arose and spores germinated into new thalli. In the case of sexual reproduction, two compatible thalli came together, plasmogamy happened, karyogamy, then zygote, meiosis, germination, new thalli. This is the flowchart for that. So with this, you can pause the video and write the economic importance as well. With this, we wind up Rhizopus.